Assalamualaikum and good morning. Today my name is Amir Abdul Rahman and my metric number is 182082, and today I'm going to talk a little bit about spectrophotometry. So the first task of this LEX 2027 task 1, I will discuss about the principle of spectrophotometry. So spectrophotometry uses a beam of light that passes through a sample, and the light transmitted is detected by a photometer. So the spectrophotometer is divided into two parts, which is the spectrometer and the photometer. So the spectrometer part is basically where light is produced and the light is focused, and then it is divided into individual wavelength, and the single wavelength will be selected, and then we move to the photometer where the light from the spectrometer passes through a sample, and the sample will be detected, and the light transmitted from the sample will be detected by the photometer, and the photometer will be passed through to a galvanometer. So basically, in a nutshell, the spectrophotometry uses light principle to show the absorbance of light of certain compound. So the next part is I'm going to talk about the structure of photometer. So structure of spectrophotometer. So it consists of a collimator, a monochromator, a slit, a cuvette, and a photometer. So first of all, a light is transmitted through a collimator, which is like a lens which focuses the light onto a monochromator, which is a prism, and then the monochromator will split the light into individual wavelength, and the slit is for selection of the wavelength suitable for the experiment. Then the selected light will be passed through a cuvette. The cuvette will be holding the sample, and then after the cuvette, the light will be transmitted into the photometer, which I explained already before. The photometer detects light, and then it passes through the information to a galvanometer. So the next part is about the absorption of spectrum as a fingerprint. So let's say compound has its own absorbance of light. For example, we have let's say a black stool. The reason why its color is black because it absorbs all light and no light reflected from it, so it appears black. And on the other hand, we have a white stool. The reason why it's white because it absorbs no light at all and reflects all lights away. So as example, in a lab, we have to determine the rate of reaction and things like that. We have a copper to sulfate, which is blue in color. The reason it is blue in color because it absorbs all color except for blue. So it reflects the blue color away, and then in a spectrophotometer will detect this wavelength, and that will determine the character or the identity of the test we're using. So I'm moving on to the next part. I'm adding as an application as a veterinary student. So in vet, we usually use a lot of bioassays. So an example of the uses of spectrophotometry in bioassays is to determine the color or the absorption spectrum of certain compound. For example, we have chlorophyll, which is already known as green in color. So we want to know why is it green. So we put in a spectrophotometer, and we have concluded that it absorbs all light except for red, yellow, and green. So this allows scientists to develop a light that can be used during day and night for the photosynthesis in indoor plants, which the light involves only red, yellow, and green color. So another application is also in determination of heart attack. So sample from a volume of enzyme sample from patient is taken, and then will be passed through a spectrophotometer to determine the concentration of the enzyme, and then the concentration of enzyme will be compared to a standard graph of concentration of enzyme, and then we can determine if the patient is having heart attack or is normal. Okay, thank you. That is all from me.